pick your subject early and become an expert at it.
Welcome to the Greener Grass podcast from BlueBird Botanicals. I'm your host, Lex Pelger. Everyone is talking about terpenes these days. These mysterious compounds are one of the most widespread classes of chemicals on Earth, and they're the molecules underlying the rich tapestry of smells and flavors that we find in cannabis. But terpenes are found in many other organisms, from fungus to flowers to trees, and there are more than 30,000 different kinds. So what's their function within the plant, and what can science and medicine learn from them? Dr. Susan Trapp joins us today to talk about her research into terpenes, and how much we've learned in the last 2 decades. So if you're curious what the smell is all about, this episode is for you. This show is brought to you by BlueBird Botanicals, to spread education about cannabis and other things on the greener side of life. BlueBird CBD oil comes from farms in southern Colorado and is grown using only water, soil, and sunlight. Go to bluebirdbotanicals.com for more info. Hello, everybody. I'm very happy to be here with Dr. Susan Trapp. Thanks so much for coming to join us.
Thank you. I really enjoyed the opportunity to come here and talk with you today.
So before we get into your research and work, did you always want to be a scientist, or was there something else you wanted to do when you were very young?
Oh, that's a great question. Yeah, I had 2 loves. So my first love was art. And to this day, I still do art. I love charcoal drawing. As a matter of fact, I'm giving away Christmas presents as art. But my second love actually was science, and I won the science fair in, I think, in 9th or 10th grade for looking at vitamin C in the common cold. There you go. Way back when.
Oh, that's great work.
Yeah. Yeah. And so I ended up in science as opposed to art.
And so how did your science career go for undergrad and learning about how to do research?
Another great question, and I hope I'm speaking to the underdog here actually a little bit. And I also teach at Red Rocks Community College right now in general biology, and so I like to. I had a wayward path onto science actually in undergrad and grad school. I started out as a history major, and Russian history actually, because I was interested in why, when I was a little kid, why everybody told me that the communists are bad. And we sort of broke that down, and it's kind of interesting. But nevertheless, so I started out as a history major and actually got a history degree, considered going to medical school, so I started taking all the courses that you needed to take to get into medical school, and actually fell in love with molecular biology way back when, here at CU Boulder. Actually with Larry Gold, who, I don't know if you know who he is, but Dr. Gold's one of the seminal professors now at CU who's had a couple of companies of his own. And so once I discovered DNA, I was hooked, and I've been on that path ever since.
So what did the research look like that you were doing with him?
I didn't do research with Dr. Gold. He was my professor.
Oh.
Yeah. But after undergrad, my first job in the sciences was at the Marine Biological Laboratory in Woods Hole. I don't know if you're familiar.
Wow, that's one of the premier research places in the country.
Yeah. So yet again, back to my background in regards to, I actually decided to give up on going to medical school, and I thought I was going to go into the Peace Corps actually and be a diplomat, right? But I ended up with a job at the Marine Biological Laboratory, to my own surprise, way back when. And I worked in a lab that was studying third messenger systems, so inositol triphosphate, way back when, so looking at neurotransmitters and how they work, and we were studying those in vertebrates and invertebrates. And one reason people go to the Marine Biological Laboratory is because you have access actually to the sea world, and so we needed squid and octopus.
Because they have particularly large neurons, correct? And there's something intriguing about working on those organisms?
That is true, and actually that research was the big giant neuron out of squid was done there at the Marine Biological Laboratory. But our lab was studying photoreception. which vitamin A is related to one of the compounds that we were looking at, which I actually think is also a terpene, now that we're getting back to the terpenes. So I'll have to look that one up. It's been a long time. But yeah, so we were studying photoreception. So we were looking at the receptors in the eyes, actually, of squid.
Wow. What was it like to be working on squid? I mean, they're such wild creatures, otherworldly.
So that's probably the first path that sent me off into the world of plant molecular biology, and how I've ended up in cannabis is because as a fresh out of undergrad, I worked on animal model systems, and I swore at that time I would never do it again. So I personally didn't enjoy using animals for that particular reason, but the research was actually very exciting. I got to get some really basic skills in protein chemistry, and I don't know if that's your background as well, but I've spent a lot of time, pouring acrylamide gels and running gels and looking at the proteins and doing Western blots and, on and on and on. And the Marine Biological Laboratory was also an incredible place to be, and probably another reason that I ended up in science. For those people who aren't familiar, they have a public talk every Friday in the summer. They have these summer talks. It's like doing science kind of in a camp, and they still do that. And it's a very tight-knit, wonderful community of scientists who come together, and they'll have very seminal Nobel laureates who will give talks on Fridays to the whole, I want to say city, but it's a town. And it was just a great way to enter the science world. It's not like that anywhere else that I know of, but that was the beginning of my science career.
And that might be a good place to pause and ask, as someone who teaches now, what advice do you have for young people who want to get into the sciences, as you're someone who fosters them?
That's a double-edged sword again, because I feel like I was the underdog. So I had dyslexia. I was a straight A student in undergrad, and from a very hoity-toity all-girls, college preparatory school. But when I went off to college, I didn't do well. And so I think I'm speaking again to the underdog, but then I'll speak to the others, and that is don't let anybody mess with your dreams. I had a GPA of a 2.4 in undergrad, and I decided to go to graduate school, and nothing was going to stop me. And I could tell you the details of how I got in, but so that's one thing I would say is don't let anybody tell you what your dreams are. On the other flip side of that is the sciences are super fun and exciting, and I think what people don't realize is how creative they are. Like, doing research is really quite creative, and but you need to, in this day and age, work hard in school, get the grades if you can.
For your career, what happened after Woods Hole?
I had the opportunity actually to meet Dr. Craig Venter, while I was in Woods Hole, and I actually interviewed with him, and this is before he was famous. He was working at the National Institute of Health, so this is in the late 80s, early 90s, and so I went from Woods Hole to the National Institute of Health, and I worked directly with Dr. Craig Venter, working on the Human Genome Project as part of a DOE Department of Energy project. So it wasn't like a full-blown study across the country. It was initially, actually, what people don't know about the Human Genome Project was that was a DOE project to actually study the effects of radiation on DNA.
Wow.
That was what spurred it. But there's way more to that. So anyway, I worked at NIH for, I think, 2 years on the pilot to the Human Genome Project. And at that point, I absolutely 100% wanted to go back to school, get my PhD, and so I did.
Can you talk about what the technology was like then for sequencing? Yeah. Because it's been such a revolution from that time to now.
Yeah. I don't know if this is in a book, but I've always wanted to write this book. I really believe that part of there was a lot going on at that time, But so in regards to your answer, we used to have to save all of the data on like magnetic tapes that were large enough, not even a CD, and it was everything about it. When we first started, they were still doing gels and radioactivity, but we were one of the first labs to actually get what's called an ABI sequencer, where you could do dideoxy sequencing, essentially sequencing, right, using fluorescent tags versus actually doing that with what do you call it, a radio labeled tag, right, and an X-ray. So that was like that was one big technology, but then actually really the data, we didn't know how to store that data, and I really think that kind of pushed this whole era that kind of happened in 2000. The whole that was part of it for sure, I think.
Wow. Yeah. It must have been exciting to be there.
Yeah, it was very exciting. We had people coming in all the time, like trying to take tours of the lab. I think we started out with 2 ABI sequencers. We had 4. One of them got retired, and at the time I think was going in the Smithsonian, and this is way back in the 90s. So it was exciting, and I got to go to the first Human Genome Conference as part of that. And that in itself, there were some very, seminal people who are now quite famous, like what's his name? I can't think of his name right now. Who invented PCR, polymerase chain reaction? Gary Mullis.
Yeah, who was inspired by LSD, in fact, for that invention.
Yeah,
LSD for PCR.
Yeah, he gave an incredible talk at the first Human Genome Conference. I think he showed a film in his backyard, and I don't know if you've read much about him. Mm-hmm. I read one book. So he got his kids involved, and he tried to demonstrate how actually PCR works at the molecular level using his kids, and I think pinball guns or some kind of guns in his backyard. I'll never forget that as long as I live. And I was like, 22 or 23 years old, and so I loved the talk because, again, I'm dyslexic, so visualization and explanation. But most people were like, how could he? How dare he? Instead of giving a normal, PowerPoint type talk. But yeah, so that first, if anybody hears this from that, it was a very fun and wild conference.
And out of curiosity, what was it like to work with Dr. Venter?
Dr. Venter has always been, actually he's dyslexic as well, believe it or not. He's always been a visionary, and the book I would like to write in that area is, does the man make the science, or does the science make the man? Because he's always been, he's very flamboyant about how he talks and what he presents. And, I think there's been, I don't think there is now, but maybe in the early days, a little bit of controversy because it's like, promise big. But he actually was able to deliver. And so I find it quite fascinating, in terms of famous scientists in the world, right? It's a great question to ask, just like Watson and Crick, were they there at the right time, or was, and that's something I do teach, we do talk about Watson and Crick and the ethics behind it, and did they deserve the Nobel Prize? And so it's a fascinating subject of, right time, right place, or, good science, great brain. Right, I guess, and that brings us. We can bring that back into cannabis at some point because, right, we're in the very early stages of all the research and the science in this field, and a lot to be discovered and researched.
And so, actually, before we get to cannabis, I'm curious how you got into it. What about your higher degrees then? Getting into those graduate programs. Yep.
So again, this is the wayward track, and back to your former question, what I would suggest to folks who are interested in going into the sciences, the earlier I think you figure out what you want to do, and honestly, if you want a really exciting, straightforward career, it's just pick your subject early and become an expert at it, and it just is going to be, if you want a stable research career, that's the way to do it. I have jumped all over, but so when I decided to go to graduate school, the only thing that I really was interested in regards to what I did is I wanted to make sure I didn't have to sequence by using radioactivity. Honestly, that was it. Since I had already been spoiled by this really cool DNA sequencing, I wanted to make sure that I was in a lab that did something like that. And the other thing that I was interested in at the time is I was actually interested in natural products and considering, as opposed to going to medical school, going to homeopathy or some kind of a naturopath. So I've always had an interest in natural products and herbs. And so in a way, it doesn't surprise me that this has been my path. I ended up working with an organic chemist who was a terpene expert for my PhD, and he is still to this day seminal in the terpene world. But what he studied were fungal terpenoids, which is interesting because I think for our listeners out there, most people think that the cannabis is the terpene world. And so he was studying some fungal terpenoids that are very toxic, and some are in molds. There's a whole world of terpenes, even in the fungal world. And so someone had approached him, I think in the late 80s, from Brazil, who had cattle that had eaten a plant that they thought the compound in it, so these cattle were having what you call mycotoxicosis. So they were sickly ill from eating something, and when they tried to figure out what that was, they determined that it was a terpene called a trichothecane. Actually, myrothecium was the fungus, and so they contacted Bruce Jarvis, my advisor at the time, because he was the actual, I think, international expert or only expert in the world of fungal terpenes, and they were like, hey, these plants are producing this, and from all of the papers it looks like they're only produced in fungus, but we have found them in plants. And so my project actually was to determine whether these were fungal terpenoids or plant terpenoids. And my advisor had already proposed, a lot of research had already been done that he had done before I got there, had proposed that a horizontal gene transfer had occurred where the whole biosynthetic pathway to produce these terpenes had been transferred from the fungus to the plant. And that was my, what I call somewhat esoteric PhD research, which was actually quite fun at the time, to tell you the truth. But yeah.
And that's wild. Can you just explain a little bit more about what horizontal gene transfer is and how that would occur?
Yeah. So the idea of horizontal gene transfer is, so you're thinking a number of genes, or in this particular case, it would have been a whole biosynthetic pathway. So that biosynthetic pathway has all of the genes that encode for gene expression, which means when they are expressed, you get these proteins. And those proteins typically are enzymes that are doing things to actually produce the compound. Like even, for example, THC, it starts out with a very simple compound, a C5 carbon compound, and these proteins, which are enzymes, actually add little compounds to them, right, to build it up into this big complicated compound that everybody now knows as THC. And so what this was proposing is the same thing, where you have that whole biosynthetic pathway, which means you have probably anywhere between 10 and 20 genes that they have to be most likely near each other on the strand of DNA or on the chromosome, and they get somehow transferred directly through, there are a number of mechanisms for this, to the plant or to some other organism. And at the time, and I haven't kept up with this research, but at the time I think there were only, because I know this was in my thesis, there were only like 3 examples of horizontal gene transfer that had occurred like this. But it can happen in a variety of ways. You can have it in regards to recombination, but that would be when it's when the cells are the same, so they're from the same species. But you can also have what you call gene jumping, and so the genes can get spliced out and somehow put into another genome. All of that is very rare, and I think it is still very rare, but I think it does happen. As a matter of fact, the reason I picked the place where I did my postdoc, which is the Institute of Biological Chemistry, was based on this same idea. There was a paper out there that said, I think it was going in the opposite direction, if I remember correctly. No, it was from fungal to plant. So Taxol, which is also a terpene, There's a drug called tamoxifen. That's Taxol, which is a complicated terpene. It's a C20 terpene compound. Maybe we'll get to that. But initially, they identified that in some fungus in the hot springs up in Glacier National Park. Is that right? No, what are the famous hot springs?
In Yellowstone.
Yeah, thank you. Yellowstone. Yeah. Yeah. And so I went to this postdoc specifically to continue on this path of studying horizontal gene transfer, and how that occurred, and because I had not actually identified that in my PhD. It turned out to be much more complicated. I think it still hasn't been identified yet, and I could tell you details, but one of those details is that way back when. So this is dating myself because I think this is a good 20 years ago. Plant molecular biology was hard to do, and plants, as you probably know, have typically bigger genomes, not always, but because we have the model system of Arabidopsis, but a lot of these plants, especially things like trees or wheat, they have they mix their genes or their chromosomes a bunch of times, and so you can have a much larger genome than you normally would. And so they have always been traditionally difficult to study. I think they're much easier now in the cannabis world, but way back when that was difficult. And so a lot of the techniques that I had learned as any young graduate school student would in terms of southern blots and molecular biology and trying to identify these genes using PCR were all set up for much smaller genomes. The reality is I'm pretty sure if I went back and got to do this research now, if those genes are in the plant, I could easily find them. The techniques just weren't there back then.
Wow, that's wild. That the terpenes are being jumped around like that.
Yeah.
And I wanted to ask you
if indeed they have, because it turned out even with Taxol that initially they found it in the fungus, but probably so Taxol is derived from the Pacific yew tree. It's a conifer tree, and essentially I think they just found it in that fungus, and there was some kind of symbiosis going on, and that's what I didn't say about my PhD as well, as trying to determine, did these genes actually get transferred, or was there some kind of endosymbiotic relationship going on where the fungal terpenes were somewhere in the endosperm or whatnot. And so same thing with this other system, Taxol, and it did turn out that they're actually produced in the conifer tree. And I don't know if anybody's, they found them in this fungus, or in the hot springs, sorry. But I don't know if they were actually derived from that. So it's still kind of an open-ended question.
Wow. And that's the fun part about science, is it's just so easy to find we don't know how that works. Maybe you'll figure it out.
Right. And then it boils down to funding.
Now, I wanted to ask you about terpenes in general, because I think so many people just know about them because of cannabis. But they're such a vital part of what makes up plants and life on this planet. And so when you're teaching students, how do you begin the explanation of what terpenes are and their complexity and diversity?
Yeah, another good question. So actually I teach biology, so I don't really teach about terpenes, but I hope I will this year, maybe to give a little lecture. But really, the almost every seminal paper when I was doing research in terpenes starts out with terpenes, quote, are the largest class of natural products in plants, period. And that's usually either the first sentence of any publication or research paper I've ever written, and in that whole field, at least when I was in it, and or in the first paragraph. And it's true. And so the place to start is, literally making that statement, and then you can break it down, because what are natural products? So I think a lot of people, even in the cannabis industry, probably aren't quite understanding what a natural product is, right? And so at least scientifically, natural products are all the compounds. Actually, when I was in graduate school, we considered them the waste product of the plant. So when I started graduate school and all the research papers, terpenes were, you'll see references to, these are just compounds, we don't know what they're for, they're essentially byproducts or waste products of the plant, but they turn out to be medicinally important. And so typically natural products such as aspirin, which I always get it confused, which I want to say is from the birch tree, but I think it's from the willow tree, right? So those are natural products. So a lot of our pharmaceutical drugs in this state in age actually come from natural products over centuries and centuries and centuries. And then we've discovered a way to actually isolate those, and then in the modern world, you end up chemically synthesizing them in the lab, like something like aspirin, and that's what gets, delivered to us as pharma. But anyway, so I sidetracked. So you start there, talk about natural products, explain that these are these compounds that are produced in the plants. Typically, natural products are considered what we call secondary metabolites versus primary metabolites. So primary metabolites are compounds that the plant, or even ourselves, produce that are essential for life. So sugar, for example, is produced by a plant and essential for food for the plant itself. Versus secondary metabolites, these are considered, again, not necessarily the waste product because we've moved beyond that, but they are not needed for or essential for life. So then it's like, well, what are they for? And then, and so if it's a 2-minute lecture instead of a 15-minute lecture, then the next level is that you have these secondary metabolites, and what are they for? And what I usually say is that they're essentially the immune system of the plant of sorts, because a lot of these compounds, terpenes, even cannabis, they have that smell, so they can have antimicrobial, anti-pesticide, but they are compounds that that are protecting the plant for a variety of reasons. That's my short, skivvy beginning lesson of terpenes.
And I get there's 30,000 of them that have been identified.
Right.
At least when you pulled together your paper, which we'll link to in the episode notes.
Yeah, as a matter of fact, I went back to double check that myself, and it's interesting. So that says a little bit about debunking the whole. I essentially went back and just checked, just briefly, how many terpenes are there, and the internet, like whatever source I got, said 20, and I know that mine from my paper back in 2000 is accurate and it's referenced, and it was at least 30,000. I'm sure there's a few more, but yeah, so it's around 30,000 terpenes.
And you called them an immune system, but they can also be a communication system between plants. What are some of the main ecological functions that you've seen terpenes used for?
So the one that I'm familiar with, although I didn't do that type of research, is this bark beetle infestation. So the lab I worked in my postdoc was a big conifer. Primarily conifers are scientific. We call them gymnosperms. So we were a big conifer laboratory because we were studying Taxol, which is derived from the specific yew, which is a type of conifer. And that was one of the big projects in the lab. And so one of the projects that we were studying was bark beetle infestation in the conifer trees. And so that is an interesting, I used to talk about this in terms of essentially chemical warfare going back and forth between the tree and the bark beetle. And it's quite cool. So what happens is if you have an area of trees that are under stress, potentially due to drought or whatnot, like not enough water for some other reason, they are susceptible. And so when they're susceptible, the bark beetle, that's when the bark beetle is going to start to come in and try and bore a hole. And so the first level of defense from the tree actually is when you bore that hole, then the tree will actually produce, which is a terpene, they'll produce a resin, which is a diterpene, and they'll surround that bark beetle and essentially just kill it. And that resin is what turns into amber, right, which is a beautiful piece of jewelry. Yeah. And so if it ends there, it ends there. But typically, when it's stressed, what happens is that bark beetle then actually also has pheromones that are also, there's a pheromone called ipsidiol that is also a type of terpene. So not only do plants produce terpenes, but so do humans, so do fungus. They're just a huge, large class. And anyway, and so these bark beetles will essentially send out a message, hey, here's a stressed tree. come on over and feed with me. There you go, a nice little rhyme. I didn't mean it to be a rhyme, but anyway, yeah. And so then you have a number of bark beetles, and then it becomes kind of a chemical warfare between the bark beetle and the tree, which is pretty fascinating.
And so with the bark beetles, you mentioned that this was a diterpene. And so for the chemistry of these, can you talk about the differences between the monoterpenes and the diterpenes and the sesquiterpenes?
Yes, absolutely. So again, these are a class of compounds, just like studying sugar. You can have a monomer, a monomer is just like a single unit of a sugar compound, the simplest sugar. But you can have monomers built into a polymer, so multiple monomers, right? Multiple units of sugar. And when you do that, like for example, that's how you get the compound cellulose, right? So terpenes are essentially the same way. You build up, you start with what we call an isoprene unit, which is a C5 carbon compound. And the ENE, when you say isoprene, the ENE stands for what we call a double bond. What happens is this C5 carbon compound, this isoprene, builds in what we call a head-to-tail fashion, and so they build on themselves with C5 carbon compounds. So you start out with an isoprene unit, which is a C5, and then if you have two of these and they get bound together without going into the details, you end up with a C10 compound. So a C10 compound is called a monoterpene. So it's just one terpene. So you have an isoprene, which is the C5, but once you have a C10, you essentially have defined something called a terpene, which is a C10. So that's a monoterpene. So then if you build on another head-to-tail fashion, another isoprene unit, you end up with a C15, and that's called a sesquiterpene. And then if you add one more, that is called a diterpene. So a diterpene has 4 isoprene units built together into a C20 carbon compound. And then in each of those classes, essentially, you can have what we call a linear compound, or a compound that's a long chain, essentially. So we call that acyclic. Or you can have a linear compound then where you have an enzyme that will actually take this linear compound and turn it into a circle or a ring, and that's called bicyclic, or cyclic, sorry, a monocyclic. And if you have 2 of those, that would be 2 rings formed, then it would be called a bicyclic ring. And so you can kind of divide each of those classes of monoterpene, diterpene, sesquiterpene, diterpene, did I get them out of order, right? They have smaller classes because they have a linear form, a cyclic form where they form a ring or a couple of rings. And then this pathway is the reason that there are like over 30,000, and maybe, eyes are starting to like, instead of sugar plum fairies, you're, like having visions of little terpenes in your eyes in the cannabis world. But the reality is that you can go up to, I think, a C20, C40. So some of the bigger compounds, precursors like for hormones, some of those are actually from that same terpene pathway. Hormones like cholesterol, testosterone, I think those are C40 compounds. I can't remember. So it's a big class of compounds.
How do those shapes affect how the molecules work?
That's a good question. I'm not sure I'm the expert on that, but so it's just dependent. So in the world of cannabis, I mean, the compound shapes are important no matter what, and that's really a big deal in drug discovery. That shape is the reason it's important is because of how it binds to essentially the receptor or whatever it is that it's doing. So it's a compound. In order for that compound to do something in the system, right, it has to either bind to a receptor, an enzyme, or a protein or whatever, and some kind of function occurs, right? So for I don't know, let's see, I'm trying to think of like linalool. Linalool is the compound that you smell in lavender, right? And I think that's a monoterpene. And in order for that to have an effect, right, it has to bind to the receptor. And so that shape of that compound is important because receptors are proteins, and proteins, some proteins are enzymes, and some of these enzymes are important for activity, right? And so you have to have this compound bind into that pocket and do its job. I don't know if that was a great explanation, but
That's a fascinating part about terpenes is the huge array of effects that they can have.
Yeah.
And one of the things you've written about, the evolution of how these occurred. And I was wondering what you could say about how these terpenes might have come into existence and diversified into what they are today.
Yeah, so that was a seminal paper probably a good 15 years ago. So way back when, this is still in the early stages of what we call bioinformatics or computational biology. And so what I did back then, and it really just, my postdoc advisor at the time asked, hey, how many introns do these compounds have? And at the time, so what we were doing is he was looking at the first enzyme in the pathway, which is called Terpene Synthase. And this enzyme is responsible, and it's actually important in some of the studies that are going on in the cannabis world, but it's essentially the first committed step in the pathway. So, for example, if you're trying to make a monoterpene, you need this synthase in order to take that linear compound and to synthesize it. And so we call that a synthase because it cyclidizes or turns it into a ring. So it takes the linear compound and then turns it into a ring. And so I looked at a number of terpene synthase genes way back when that were available in both angiosperms and gymnosperms. So angiosperms are the flowering plants and the gymnosperms are the conifer. And again, this lab I was in was all about conifer trees. But they were trying to actually isolate the crystal structure of the synthase. And so anything and everything that revolved, this is how research works, around that they wanted to know. And so I went to town doing a bunch of PCR trying to understand, what I called the gene architecture or gene organization, the gene architecture of these synthases. And at the time, I think there was probably 6 or 7 of those genes available, primarily from our lab, and I think another lab at the time had a couple of them in, some angiosperms. And then the beginning of the genome, other genomes besides the human genome was starting to explode. So there were some information on Arabidopsis synthases. And what I was able to do was to take those genes, line them up in a program where you can look at their sequence, essentially the nucleotide sequence, and begin to identify by using programs where the introns were and where the exons are. And yeah.
Can you define introns and exons?
Yeah. And so when you're looking at a gene, you get this mRNA transcript. You have a gene, and in order for it to be expressed into a protein, you go from, we call it the central dogma of DNA to essentially RNA or messenger RNA, and then that gets expressed into what we call a protein, without going into any of those details. But so the initial mRNA transcript has these regions, what we call exons, which are actually important. They essentially hold the code to translate this into a protein. But these genes also have these regions in there that are called introns. And the introns, in the old days, we used to think they were just nonsense pieces of DNA with no purpose, but that's not necessarily the case. However, these introns actually get what we call spliced out. So you can imagine if you had a long string, just a long string, and put a little golf ball on the front, right, and call that the initial part of the gene, and then you have the string that goes all the way down, and then you have we put like a little, I'm making this up as I go along, pink ribbon on the end, so that's what we would call the 3' end. But in between, so where that golf ball is, and just say the first 4 inches, we would call that region an exon. And if this gene had introns, the next region would have what we would call intron 1. And so then, the first 4 inches is an exon, the next 2 inches, let's say, is an intron, the next 4 inches again is an exon, and the next 3 inches is another intron, and then you have one more region of an exon. And so our what's cool about DNA, or just the machinery that's in all of our cells, is we have the ability to actually splice out those introns because they're not needed to code for the protein. And if they're in there, they actually mess things up, and then you end up with a protein that doesn't work. Now, that was a very long explanation, I know, but it was one that I explained orally without any visual tools.
And I think one of the interesting parts about that is, as you said, introns used to be seen as nonsense and called junk DNA.
Right.
And now it seems like they, I've heard the phrase that nature is an excellent housekeeper, and it's not going to keep things around that are specifically junk, and that these are often control systems for what's happening with the rest. And the interesting part of that, I assume, for your research is you can see the evolution from wherever this might have evolved initially, and how it branched down to all of these different plants, and what changed, and what's vital, and what parts can be shifted around.
Yeah, and so in terms of the evolution, that paper was all based on literally looking at this terpene synthase in conifers and angiosperms, so conifer trees, pine trees versus flowering plants. And the idea is, or the going theory, at least back when, is that conifers were older than flowering plants. And so I was able to kind of piecemeal together the conifer terpene synthases were much bigger with many more introns than the flowering plants. They had fewer introns, and so the theory kind of goes behind talking about the evolution of these is that initially they were genes that were important, back to this idea of primary metabolism. They were important for plant growth. The gibberellins are also terpenes, and they're essential for life and important for growth. And this paper, I proposed that there was a duplication, so this gene somehow got duplicated. And so now you have two copies of them. One copy remained important for primary metabolism, essential for growth, and the second one actually spurred off into this whole kind of set of secondary metabolites that had multiple purposes. Yeah.
And that's an interesting part about human breeding techniques is a lot of our common plants like wheat, humans have actually duplicated their genomes, so you have extra, I think wheat usually has 4 copies of its own genome, because that means there's more growth hormones in there. And it's some of those techniques which have been applied to most plants but have never been applied to cannabis.
Mm-hmm.
And so I guess I was curious, going from this expertise in terpenes, how did this bring you to this cannabis path?
So I finished my postdoc in terpene research, and I decided at that time that a lot of the research in terpenes was either very basic research. So again, this was like 15 years ago. Nobody ever heard of terpenes. All excited about the cutting edge technology that was going on. What became really exciting at the time was bioinformatics, or computational biology, was exploding, and so was a technique called microarray. And so instead of wanting to, take this terpene research and go create drugs for human use in a pharmaceutical environment, I decided to follow my bliss and went on to work for a company that was working with microarray, and so to bring it back to how I ended up in cannabis, I've jumped around in my career kind of in this field of molecular biology and DNA and the human genome. So from a non-scientist perspective, it probably sounds like it's very narrow, but from a scientist perspective, I actually did a bit of jumping. And so after I worked for this company called Informax that no longer exists, but was essentially doing bioinformatics kind of on a platform. They developed a whole platform so that you wouldn't have to go out to NCBI and look at those tools. You could just have it all under one roof. And after that, I ended up working for an algae biofuels company that was doing studying, trying to improve lipid production in algae for biofuels. And there's also terpenes there, actually. But and from there, I started to notice, and like I had a lot of friends who smoke pot, and they're like, yeah, cannabis have the, it smells great. Isn't this great? That's the cannabis, and they had no idea what a terpene was, and I was like, no, the smell that you're smelling is a terpene, and that didn't catch on for a while. I kid you not. And sure enough, a couple years ago, people started talking terpenes, and I was like, I really want to be in this field. That's how I came to be in this field. So I started started consulting a little bit in the cannabis industry.
One of the areas that you have talked about with me before was personalized medicine and cannabis. Mm-hmm. And I was wondering where you think that's going and what you are excited about in that part of the field.
Yeah, and so that's so personalized medicine. Actually, way back when I worked with Craig Venter, we had a postdoc who was interested in personalized medicine and gene therapy, and so interestingly enough, people have been trying to do this for a long time, somewhat unsuccessfully, and in all reality, personalized medicine is just coming to in its own in this day and age, and part of that I think is all the computation, and in the news these days we're hearing about artificial intelligence, and so the science field has been utilizing not necessarily just artificial intelligence but all types of computer programs and algorithms in order to begin to study this. And one of them is personalized medicine, and that is being able to utilize all the data out there to actually optimize or customize what is important or good for your own system. And so that's pretty complex and requires a big sample size and a lot of data. And so the same is going to become important, and probably even more so in the field of cannabis, because it's a complicated plant. Not only is it a complicated plant, but we have this thing we call the endocannabinoid system. So our body ourselves, right, has its own system, biosynthetic pathway that produces our own essential, I guess you could say, terpenes. They're not. I don't know if anandamides are they.
I think the cannabinoids are generally considered a type of a subclass of terpenoids.
Okay. Yeah, I haven't spent any time looking at those, but nevertheless, so we have a system that produces them, and now we also have a plant that's producing them, and we're having both of those in our system. And so the idea behind personalized medicine is really start to take that data and understand it, one, on how it's working, and two, utilize it for everybody on a personal level, I guess you could say.
Well, then we'll end with another question maybe you can hone in on, which is if you could either get a dream project in your cannabis consulting of something you'd like to look at, or we could give you a giant grant to study exactly what you wanted to for the next 3 years in your own lab, what would you be looking at the most with your research money?
That's such a hard question.
Good.
Good, yeah. I guess there's two areas of research, and one is really what we were just talking about, and that is taking all the data types, like understanding the endocannabinoid system and understanding the terpenes and the cannabinoids, and really beginning to understand what they're doing, sort of essentially basic research. But one approach to that, again, is essentially computation and databases and being able to, or this big field of data science nowadays. So taking all that, gathering that input data and then actually massaging it and analyzing it such that you can make inferences about how this is all working together, so that you could better predict how someone's going to respond to recreational cannabis, or how somebody's going to respond utilizing cannabis for a specific thing like seizures, because there's a lot of individual traits, I guess you could say.
Well, thank you so much for coming in to share about this, and hopefully we can check in later and hear how your various projects are going and all the work you're up to.
All right. Thank you. I really thank you. Thank you so much for inviting me today. I enjoyed immensely talking about terpenes. It's something I haven't gotten to do for a while.
Excellent. We were happy to hear it.
All right.
Greener Grass is a BlueBird Botanicals podcast. Their CBD oil supports a healthy body and a strong endocannabinoid system. They've got friendly customer service who can answer any of your questions, and the number is right there at the top of their webpage. But, per the FDA, they won't be able to make any medical claims for these nutritional supplements. That's also the reason you'll hear little about CBD on this show. There's no need for us to add more evidence about CBD when a simple Google search will give you more than you can read in a month of Sundays. So this show covers the cannabis world and more with editorial freedom from BlueBird Botanicals. Thanks for joining the Greener Grass podcast. As always, our audio alchemist is Matt Payne. The Gypsy Jazz theme music comes from Brett Van Donsel. Our beautiful bird sounds are courtesy of Lang Elliott. And I'm your host, Lex Pelger, wishing you a bright green day.